So good morning, everyone. I know this is the panel session right before lunch, but I think it'll be a really good panel session. We have a lot of good and experienced regulatory colleagues with us. I'm going to introduce everyone briefly, but then if you want more information, you can always look at their bios, which is in the back of the program. So this is the regulatory panel. We're going to be focusing on bringing innovative products to the market within our current regulatory environment. My name is Tsokas. I'm the Senior Director at Janssen Research and Development, which is the pharmaceutical companies of Johnson & Johnson. And we have with us today Anne-Virginie Eggimann from Bluebird Bio, Ellen Feigal from CIRM, Natalie Mount from Cell Therapy Catapult, Robert Pietrusko from Voyager Therapeutics, and Celia Witten from the Food and Drug Administration. So I'm going to ask our panelists first each of them will have around 5 minutes to speak to us about what do they think our challenges and the opportunities are within this regenerative medicine, cell, gene therapy space, and what issues do we think that from this current regulatory environment, how we can move forward and work with them as an industry as a whole. So, Anne-Virginie, since you're number one, I'm going to start with you.
Okay. Great. So Bluebird is focusing on the development of gene therapy for the treatment of rare diseases. We use ex vivo lentiviral vector platforms, and we have a couple programs in hemoglobinopathies for the treatment of beta thalassemia and sickle cell disease. We also treat a rare CNS disease called adrenoleukodystrophy, and we are focusing on the childhood cerebral form of the disease. We also have an oncology program where we modify T cells using lentiviral vectors. So in terms of our opportunities, I think because of the potential transformative potential of our therapies, there is a potential for cure, or certainly a transformative impact on the patient. So that's certainly one of our major assets. In terms of challenges, we've discussed that with the group. Because we use lentiviral vectors, they integrate permanently in the cells, and so you have a long-term effect. And when you think of evaluating benefit-risk, there's going to be issues about how much long-term follow-up and how much long-term safety data will be required for registration. So these are some of the topics that we are thinking about, and it may be helpful, just as mentioned this morning, to have several companies come together and discuss the scientific approach to do integration site analysis, what is the best approach to evaluate these risks, and from the regulatory perspective, what is going to be required at the time of BLA in terms of follow-up for patients and to evaluate these risks. And another challenge, which is a good challenge to have, is as we're thinking about accelerating our clinical development based on outstanding efficacy, every time we sort of map that out, what comes out is actually that our CMC manufacturing, the product development, becomes rate limiting. And so we look forward to having discussions with the agency on and I think, Celia, you mentioned that this morning, on how best to interact with the agency to have potentially more frequent discussions with regard to CMC, to be able to perhaps agree early on about things that could be done post-approval if the clinical development is really accelerated. So these are some of our challenges, and in addition to a more classical challenge, which is conducting clinical trials in rare disease indications with very few patients. But that's, I would say, not specific to gene therapy, but we do struggle with that to a certain extent.
Thank you. Ellen?
My name is Ellen Feigal, and I'm Senior Vice President for Research and Development at the California Institute for Regenerative Medicine. That's the $3 billion state agency that really was put into place, as you heard about earlier this morning, in 2004 to really advance stem cell-based science towards therapies that could really make a significant impact on patients who have debilitating diseases and injuries. My role there is really to help advance the programs that are entering into the preclinical and the clinical space and help shape them so that they can move forward into the clinic. At this juncture, we have about 90 projects that are in what we call the translational space. And of those, we have about 27 that are working near term on getting into the clinic. And we have 18 right now that are funded through clinical trials. And this year, we will have, before the end of the year, our expectation is 10 active INDs with active clinical trials that, and some of which have already started, some of those programs have already started enrolling patients, about 6 to 7 of them. So we really are trying to advance what has been a very complex scientific, regulatory, technical, business challenging type of approach into the clinic. We've had very, I think, very productive, very good interactions with the regulatory agency, with the US FDA, and with Celia Witten in particular, in terms of thinking about the pioneering path to bring these types of therapies forward. In addition, we work extensively with companies, primarily small companies, and a lot of researchers in the academic sector. So there's not a lot, a ton of resources for them to be able to move things forward. So as an agency, we're really working on those aspects of the development program that have the highest amount of uncertainty in terms of moving through, so that we can hopefully reach an inflection point where we're gaining enough data so that it will be attractive to perhaps larger biopharma, biotech investors to move the program all the way through to registration and to commercialization so that it can reach patients, which is obviously our ultimate goal, is to accelerate therapies to the patients. So in considering all that, we actually there's a variety of different regulatory challenges, but in trying to think of what might be unique to the United States versus what might be globally, because many companies, many sponsors are thinking about these therapies as global markets, not just particular to a state or to one country. We held a workshop back in September of 2013 where we brought regulators from all over the world together into one place to talk about what the regulatory challenges might be on the cell development pathway, and then talked about those actionable items where there might be a way to mitigate the uncertainty of the cell development pathway. And there were 5 areas that we came up with. One was the area of donor eligibility and suitability of cell lines for going into first-in-human clinical trials, but also going into commercialization and trying to get better clarity on what's called the exemption or exception pathway for those cell lines that were derived prior to May 25th, 2005. The second was really about the comparability of the types of studies you need to do between master cell banks as you're working on the manufacturing issues. The third was really trying to get a convergence in regulatory guidelines for some aspects of cell development that are common no matter what country you're working in. And these are the areas of assessing for tumorigenicity and assessing for biodistribution. And right now, different regulatory jurisdictions have different guidelines. And scientifically, we're not really sure that makes a lot of sense. So seeing if we could get some convergence or some harmonization on those issues. The fourth was really thinking about really a challenge to investigators, whether you be in small biotech or whether you be in an academic center, to actually share some of the information you're learning about the methods, about the assays. Those are things the regulatory agency can't share. It's confidential, proprietary information. But if we really want to accelerate the field, find some way that we can be able to share that information. The other is sharing of information more broadly and disseminating results more quickly from our early phase clinical trials, so that hopefully we can prevent people going down the same blind alley as they're going out through things. And then the fifth is really finding a forum. Earlier this morning you heard from the Japanese ministry about the very innovative approach they're taking with regenerative medicine, having a very expedited pathway that's tailored to regenerative medicine, You heard from Dr. Witten earlier about some of the expedited pathways in the United States. So thinking about some form or way we can share, so how's that working for you in terms of trying to get this investigational, innovative type of therapy forward? So those are the 5 issues that we see.
My name is Natalie. I'm the Chief Clinical Officer at Cell Therapy Catapult. And the Cell Therapy Catapult is an organisation which is a not-for-profit company, really set up to grow the cell therapy industry in the UK. And we collaborate both nationally in the UK with academics and small companies, but also globally with people for them to bring.
Working better? Okay.
Yes.
Yeah, okay. So at the Cell Therapy Catapult, we're a not-for-profit company, and our role is to build a cell therapy industry in the UK. So we collaborate nationally and globally, really encouraging people to accelerate development of promising therapies and to bring their European development into Europe through the UK. So as the European representative on this panel, I'll talk a bit about European regulation and some of the challenges and also some of the things, important things that are happening in the European regulatory framework that I think people in this audience ought to be aware of. As Ellen and Anne-Virginie said, we all start to look globally, and people will be bringing their product development into Europe if they're not there already. So most of these cell and gene therapy products in Europe are regulated as what are called advanced therapy medicinal products, so-called ATMPs. And that regulation was drafted in 2007 in the EU and set out the fact that these products need to come through a central licensing process in the EU. So the European Commission have recently conducted a 5-year survey looking at the last 5 years since the regulation was enacted and asking people for what challenges they've actually come across and what the impact has been of that regulation. That report was published in about March of this year, and it's on the EMA website. People want to look at it. And it contains actually 8 recommendations for the Commission to consider for change to the regulation. So within those, I won't go through all of them, but there are a couple which I think are really important to this field. One is around looking again at the provision for what's called the hospital exemption clause in the EU. And people working in the EU may be familiar with this. This was an exemption for marketing authorization for ATMPs that are made to fulfill the needs of an individual patient and their particular need under the responsibility of a physician in that member state. And the idea behind the hospital exemption clause was to make sure that patients could get access to innovative medicines ahead of formal licensing. The way it's been enacted in different countries means that, unfortunately, in some countries, it can provide a disincentive to formal clinical development because hospitals are effectively making unlicensed products. And when a licensed product becomes available, the Commission now want to look again at how that hospital exemption clause should be enacted and the conditions under which it should take place. So that is a really important element, and I know people developing certain products are being impacted by how the hospital exemption is being used. Another area they looked at more generally is a very important general statement the Commission made, where they said actually looking at the impact of the regulation, the impact has been that there were about 32 products on the market before the regulation came into force. Now we have 4 approved products on the market. So clearly there's been a big attrition of products that were previously on the market to those which are now licensed. And they've said they want to look again to make sure that the European framework is proportionate for these kind of products and actually adapted to the special scientific needs of these products. And so we can expect them to look again at things like manufacturing requirements, CMC quality packages, as well as clinical safety and efficacy information. Another area that's kind of related to that is the changes that are going on in Europe in terms of accelerated approval programs. We've heard already this morning from the US and Japan about their regulatory frameworks. So at the European level, the EMA has been looking at this, and people may be aware that they're introducing a new adaptive licensing framework. So at the moment, the types of approval you can get are a standard or accelerated approval, a conditional approval, or an approval under exceptional circumstances. With the adaptive licensing, they're saying that any of those types of approval could come under an adaptive license, which means that prospectively during your development program, you plan maybe a particular niche population of patients who will get approved first, and then you'll broaden your indication out post-approval. So it is an important, different way of looking at development. And a number of pilot studies, the commission opened for people who wanted to bring forward their products as potential pilots. Those have now been selected, and over the next year we expect to see some readout from those pilots, which will give much more substance to how adaptive licensing will work. And then the last thing I wanted to highlight is actually in the UK itself, we've been looking at how we can accelerate innovative products and innovative medicines. And the UK MHRA earlier this year introduced their new Early Access to Medicines scheme, which is a two-stage scheme. The first is getting what they call a Promising Innovative Medicine designation, which in some ways is similar to the Breakthrough Therapy designation in the US. And actually the first therapy has just been awarded a few months ago, actually about 6 weeks ago, their Promising Innovative Medicine, and it went to a cell therapy product, a dendritic vaccine from malignant glioma. So the idea, having had a Promising Innovative Medicine designation, is that then you can then apply to get an early access scientific opinion, which would allow you to start treating patients in the UK ahead of your central European marketing authorization. So those are some of the things I wanted to highlight in terms of what's going on in Europe that may impact this field.
Okay, can you hear me? I'm Bob Patrusco. I'm Senior Vice President of Voyager Therapeutics, a gene therapy company that was formed in February of this year. And the company is using the adeno-associated virus serotype vector platform to develop therapies for neurodegenerative diseases. And there's a significant unmet medical need for these conditions, such as late-stage Parkinson's disease, ALS, and Friedreich's ataxia, and there are many other diseases. And the issue has been that many of the small molecules and other therapies have been largely ineffective in treating the underlying condition for various reasons. Primary among them is the inability to penetrate the blood-brain barrier. The approach that Voyager is taking is that we're using convection-enhanced delivery of injecting the product either into the brain directly or through the spinal column with the intrathecal injection. And the opportunities are many in that the products will stay in the area and will be relatively isolated to cut down on the chances of systemic immunity or off-target places such in the liver and other parts of the body. So that's very important. And we're focusing primarily on diseases initially that are monogenomic, so that we're looking at either at replacing defective genes or knocking out other genes, such as with knockdown therapy for inappropriate or ineffective proteins in the body that will cause problems, such as with ALS. And so there are numerous opportunities for making a true difference in these patients' lives. With gene therapy and this type of a product, it basically will be one injection. And hopefully the expression of the gene or the knockdown gene will occur for many, many years, perhaps a lifetime of an individual. So it's the convenience of one injection for a lifetime. So there are many opportunities in this area for these unmet medical needs, looking at curative outcomes. With those opportunities come a variety of regulatory challenges. And among them is the concept of one injection for the lifetime of the patient. So the traditional standards of looking at therapy is single dose response studies. What is the maximum effective dose, minimum effective doses? Can you use normal volunteers? Cannot. So you cannot use normal volunteers, would be in patients. And with the risk of potential immunity to the viral vector after initial administration or to proteins that haven't been available in that particular patient being reviewed as foreign, so that readministration can be highly problematic. So that for patients involved in these trials, once they volunteer, they receive a dose of the product, basically they are prohibited from receiving further injections or possibly other products. So those are the challenge we face. Also, the preclinical work, the support of animal studies, pharmacology to support the therapy, in that you have animals that you treat for the acute, subacute toxicities. When is enough of the animal data to support going into humans for continuing the therapy? So these are some of the issues we face. The other is with some of these neurodegenerative diseases, the effects and the onset are long-term. It may take 2 years before you may see a difference. And we're just learning about the potential biomarkers, genetic expression, gene expression, what is relevant, how much of the protein is necessary to correct and prevent the diseases from progressing. So the time frame is much longer in many of these conditions. So we're looking at how we can facilitate that process and continue to develop products in this interesting area.
Thanks, Bob. Celia?
Yes. So, I'm Celia Witten. I'm the Office Director of the Office of Cell Tissue and Gene Therapy at FDA CBER. And I think probably most of you were in the audience during the last session during which I spoke. So I don't want to repeat anything that I said there, but I'll just build on one of the comments I made, which is just that, just to talk about the need for information sharing and for the stakeholders community as a whole, by which I mean not just industry, but industry and the academic scientific community as well, to think about the best ways to share scientific information or develop scientific information and test methods to fill in some of the scientific knowledge gaps that I think are challenges impeding the development of products. And it's a new field, regenerative medicine, and multidisciplinary. Since the scientific advances build on past work, it's really critical for all stakeholders involved in product development to be able to rely on scientific literature and knowledge as the foundation of their further innovative product development. And I think that's sometimes made a challenge because of the way things are reported, perhaps inconsistently, or also because there are some things that just aren't shared in the public domain. So an example of inconsistent reporting would be the example I gave about MSCs, that it's really hard to compare or to know what you've learned when you clearly are looking at a heterogeneous, the literature is pointing to something heterogeneous. But I'd say on the other end, test methods, it would be great to have some standard test methods, and whether it's for things like vector integration or anything else, the more that the stakeholders can work together to try to figure out the best way to develop standard test methods or ways of information sharing that can be understood and interpreted by regulatory agencies, the better, I believe. So that's a challenge to the field. From our end, one of the things we're doing, and I don't think this is all covered by the notion of consensus standards, but I will say that there are consensus standards organizations that work in this area, and we had a workshop probably about a year ago, to bring a number of stakeholders together to talk about development of consensus standards, and I think that is one area that would possibly benefit from more attention by more stakeholders in the field. Because as it is, every time there's a question, each sponsor is providing their own answer to that question, their own test method that they've developed, that they've explained, that they've validated in some way. And so we're looking at different approaches to address the same problem multiple times, and I think the field might benefit from some collaboration about those kinds of issues. So.
Thanks. Okay, so I'm glad that the panelists shared their thoughts of our challenges and opportunities. We will open it up for questions from the audience, but I just wanted to start off because I've been hearing a lot individually as I chair the Alliance for Regenerative Medicine Regulatory Committee, and the accelerated pathways often comes up, right? It comes up not just from the U.S. perspective, it comes up globally. I know the UK, but also the pilot program that the EMA is running. So how can we, from a regulatory strategy perspective, we work with our colleagues. We want to bring products forward faster. So our clinical teams work, and they have wonderful programs to accelerate the clinical program. And yet our manufacturing colleagues are always saying, wait a minute. We won't have adequate supplies or supplies that actually meet phase 3 commercialization in your timing that you're proposing with this accelerated path. So I wanted some of the panel's insights on potential ways that we might be dealing with it, or thoughts on how to best address. So I don't know, we can...
I'm not sure if you're asking a pragmatic question or a regulatory question.
Well, I guess it is a pragmatic question, because from a regulatory perspective, you're right, we have the guidance, we have the regulations. So this is more a pragmatic, and from your experience in regards to developing products from a regulatory strategic perspective. So not necessarily the guidelines, but just in developmental purposes.
So you want the regulatory person to talk about it from a pragmatic perspective. Okay, I understand. So, no, I think that's a totally logical question. And I will repeat some of what I said earlier, which is that it's certainly true that if the clinical development is proceeding apace, there could be a misalignment with the readiness of the CMC, and so you want to start thinking early about the critical quality attributes of the product and look at what your plan is for demonstrating product comparability and some other basic things like stability. How are you going to look at some of those things? Our staff will certainly discuss those with any sponsor, and in particular, if someone has breakthrough designation, that's something that they could come in and say, we want to talk specifically about that issue, of planning early for our development of our phase 3 product. Well, we talk to other sponsors about that too, not just breakthrough. So pragmatically, I'd say there's certain key issues you want to think about early, especially because those are the ones you're going to use for comparability. You want to nail those down.
So moving ahead with the product that you have, let's say at a phase 2, but having the certain criteria that you need to do the testing for comparability, so that if you are approved earlier, accelerated, you can then address changing the process.
And I'll be happy to have this problem come up, by the way, of where somebody is, going so far with their promising product that they want to discuss these things with us.
Yeah, I think obviously the comparability question is absolutely central to be able to make those changes to the product. The other thing I would say is I think all of the agencies are very willing to meet and give advice, and people should take note of that and actually make use of that to actually go, because there are different requirements in Japan. This is emerging now in what Europe will require and what the FDA requires, and whilst there is an element of convergence, there also are differences in what people need. So it's really important, I think, to go and have those conversations with the regulators at an early stage, so you actually know what's going to be required for each jurisdiction.
I think in terms of some of the projects that we're working on, it's still early in its phase of getting into early phase clinical trials. So there's it's all done under GMP, but there may be some bioprocessing development and other changes that will need to be made. So for us, some of the things are sort of a pragmatic strategy issue, is when to institute those changes. Do we wait to see a signal of biologic activity or some preliminary evidence of clinical before we invest in the very expensive bioprocessing development? Or because if there are accelerated timelines, do we just take the risk and do it in parallel? There's no right or wrong answer to that. It really is a judgment call, but those are some of the issues that we're facing.
I think it's important as we go forward to learn and have an understanding of those critical manufacturing parameters that really make a difference. And we're in the early stages now, and we need much more scientific information. As we move forward, one of the concerns would be a designation that it is a new product, and that causes many issues and problems going back and repeating pharm tox studies and can delay the program substantially. So I think as we move forward and learn about the comparability studies and critical parameters, we'll be in much better shape to move that forward and keep the supplies going for the patients in the treatment programs.
Yeah, on our side, we are certainly hoping to benefit from breakthrough therapy status or other potentials in the EU and other regions to have more frequent discussions with the agency and earlier discussions about how can we accelerate the product manufacturing timelines. But on our side, things are quite complex. We have to validate the manufacturing of our starting materials, including plasmids, and then we have to do the lentiviral vector, and then the drug product. And certainly I would encourage sponsors to think about their clinical development and having discussion with the authorities on that. But and then as a second step, we usually discuss the CMC aspect, and what we're finding is that there may be a need to have the validation of the drug product, for example, be completed post-approval. And so that's a pragmatic question, but also a regulatory question is how, within the existing frameworks, how would that be possible, for example, to either file the validation of the drug product during the review of the BLA or as a post-approval commitment. And if it's not possible, then how can we manage to know what we're going to need to file and what could be provided post-approval? So it's an evolving discussion because you need to sort of clarify the clinical framework first. But these are certainly topics of interest.
So I think the other thing for us is has to do with the cell source. And some of our lines were derived prior to the May 25th, 2005. And so some of the issues are they're on an exception pathway. And we understand, we're grateful for the exception pathway, but we want to know how fragile that pathway is. And is it a potential pathway to get all the way through commercialization should safety be met and should real clinical benefit be shown? And so that is an issue, and the answer could be it will continue to depend, but it might be helpful if perhaps thinking of the opposite question, what would be the criteria for revoking an exemption pathway? As opposed to asking, could we get it? Maybe what are the criteria the regulatory authority is thinking about for revoking an exception?
Is that a question for me?
I think so.
I'm a little confused about the question.
The question to try and clarify it is right now for cell lines that might be on an exception pathway to go into, say, an first-in-human clinical trial.
So those are after 2005. Sorry, go ahead. These are.
So the question is, there's permission to go into early phase clinical trial, and we will ask permission at each step of the way. So the question is, sort of as the agency thought about what some of the general criteria would be to say you can no longer go to that next step.
Well, I like the way you put it the first time around, which is what would be the criteria for applying an exemption to a marketed product. Is that what you actually are trying to ask?
That's what I'd like to know, but please, whatever question is clear to answer, go for it.
Okay, so I think what you're asking is, is what for licensure. And as you know, I'm sure, there's a lot of things that if you did not follow the donor eligibility or you don't have that information, you can test the cell lines for that. But the exception would be, in terms of the current regs, I mean, there could be new diseases in the future. You're not asking about that, though. The current exception would be TSC. I'm assuming that's what you're referring to.
Yeah, that's the one where we don't have a good test.
Yeah, okay.
Yeah.
So that one we don't have a good test. So I think of course, we'd prefer not to have this question come up, but we realize that it's inevitable. And we would expect a sponsor to do the most they could at the time of licensure to, to do any testing that's available at that time or document whatever they can about, the cell source. In other words, there isn't a, we would want to know as much as we could about safety. And then I think the criteria and the exemption are fairly broad. They ask whether, that action would be consistent with public health. So I think we would look at the risk and benefit of the product, including that as part of the risk that we would look at. So that would be my, scenario for this.
Well, that's helpful.
That's what we look at.
I just wanted to add it as well on cell source. One of the requirements that people need to be aware of in the EU is what's called 30-year traceability. And this is the requirement to have maintained data for 30 years, which links your source of your product with the manufacturing process and then the patient who received that product, and also to maintain the records of product contacting materials which may affect the quality or safety of the product. This is something that, similar to we were just discussing with the cell source issue, can trip people up if they haven't actually done that and kept those records. So we have now a new template called a cell history file, which actually lays out exactly what needs to be collected and how to document it. And it's really important to consider those things, because otherwise people will be coming into Europe having these same discussions about whether it's possible ever to fill a gap for lost documentation or things that weren't collected, that should have been collected and need to be kept for 30 years.
Question?
So I have a couple of questions. Can you hear me?
No.
Not very well.
Hello? No? I'll raise my voice. I have a couple of questions. One for Robert in terms of your proposal that a single gene therapy delivery would actually help a patient for his lifetime. But what happens if that single delivery actually by itself leads to an immune response? Well, how do you shut it down if your protein is being expressed and now all of a sudden the patient, has these reactions? What is the antidote to that? How do you handle that? So that's one question. And the second question is for Dr. Witten. In terms of control, especially in this area of cell therapy where a lot of our targets are very high unmet medical need, very few options, it can lend itself to perhaps situations where we can do registration studies to understand the population and the endpoints. But is there a venue for having studies that are not necessarily placebo-controlled? You don't have alternative treatments, and it's very difficult to enroll these patients. And then the whole ethical issue about whether or not, if they are terminal, and it's very difficult to actually enroll them if they don't know that they're going to get something. Is there room for those types of controls?
So
Okay,
Bob, you
I'd
wanna go
like to answer the questions for you. The first one was around the potential for long-term immunotoxicity and reactions. And there's several points. One is by administering products into the CNS, the blood-brain barrier works the other way. Hopefully that there'll be very little, if any, that will come out from the CNS going to other parts of the body. So that risk is reduced in that way. The second is looking at particular conditions, and that these are recombinant human genes. And if a person has total lack of this gene, the risk is greater. And so those are those conditions where the benefit risk, you'd have to make sure that these individuals do not have that particular gene at all, and you may introduce that, and there would be an autoimmunity. In those particular cases, then they would be on immunosuppressives to correct that. So it's very serious about how you look at that benefit to risk. So that's what we're looking at, and we'll do the animal models, and we'll do other types of tests to focus on those kinds of things and reduce it. Now, there are other conditions where there's a relative lack of a protein, And in those cases, it's recombinant. The chances are much less that would happen. Plus, if you're in the CNS, then there's less chance it would go out. So that's how we're addressing it. Okay.
So with respect to the question for me, we've always considered, alternate study designs, and would certainly continue to do that. Our focus is on making sure that the study is interpretable. And so it isn't that, I can't say a single-arm study would be okay if there's a small patient population and not if there's a large one or anything like that. It may or may not be acceptable with a small population depending on whether the study is likely to be interpretable. I mean, to some cases there may be a huge effect that is clearly couldn't occur in nature. And in other cases, it might be rather ambiguous to interpret such a study. Like I say, a typical example of this are time to event studies are, I think, often, it depends. I wouldn't make a blanket statement, but they often are hard to interpret when they're single-arm studies. So I wouldn't, I would just say if you have a proposal and for your product and your sponsor and you believe it would lead to a study that's interpretable, which, remember, we all have the same goal, which is to make sure that the products, if they get on the market, are products that really provide value to patients. And it's not just, well, they need something, so, we're hoping this works. We all want to, I think it's a shared goal of the community to make sure that anything that is on the market is something that's going to help patients and make them better. And so if the study would be interpretable and we would be able to understand where that benefit resided, then that would be a reasonable study. But it would really just depend, so that's all I can say. And that's always the case with study designs for FDA, is it depends.
Any other questions? Okay. So I want to go back, because several of you mentioned the fact that, if we could, as an industry, work together to share information and just to maybe be a little bit provocative, Ellen likes that word, in regards to if to gather safety data, if we talk about cell therapies or gene therapies as a class of products, is there something that we could do as an industry to gather information to then support the fact that there's safety evidence for individual products as they move forward?
So certainly I know that earlier this year, FDA and ASCGT organized an event where we discussed, it was focused on gene therapy, but we discussed the best way to conduct safety studies, toxicology studies for gene therapy, and I thought it was very helpful, and a lot of people from industry and academia came and discussed the best models and the appropriateness of the models and the fact that there are actually very few models that are actually helpful. And I thought that was a great example of putting thoughts and scientific approaches together. So certainly, as I mentioned on our side, it would be helpful to think about integration site analysis and pulling data from different companies that are developing lentiviral vector gene therapy to see if there are standard ways to look at this data. It's very complex. It's a lot of data, and if you're not a specialist, it's really hard to interpret. What does it mean? Gene integrates here or there, and depending on the indications, it can lead to oncogenesis with gamma retroviral vectors, but with lentiviral vector, it's never been observed, and there's a lot of unknown, and so trying to understand this mechanism for potential toxicity would be really helpful, and I'm sure the agency could also provide some guidance as to how best to analyze the data, what to do with it, and how to interact with them based on that.
I think it's important that we do communicate and share information, especially in this area where the studies are few and the data are limited and we're in this learning phase. And there's several ways to do that, is with a group like at ARM, a subcommittee to discuss the safety profiles of the products and share that information, particularly around the safety, what's being seen. And I think it's going to require cooperation, and it's that delicate balance because we are competitors in the areas. And another way is also allowing a cross-reference to an IND for the FDA to look at the safety profile and consider that, particularly in gene therapy, if it's a similar serotype. Are there certain similarities that can be seen with that particular serotype? And we go with other vectors, and there may be less similarities with a different vector. There are currently 100, depending on biodistribution, targeting, and detargeting. So there are several ways to do that. But I think it's going to be absolutely essential that we cooperate and help determine the safety profile of these products.
I just would like to clarify. I'm sure these are all good ideas, but they're a little bit farther along than anything I was suggesting. What I was really saying was that just, taking a step back, I'm not sure it's just the product development community, it's the academic stakeholders too. But I was really referring to perhaps an earlier stage, for example, development of test methods. That is something that, there isn't in common. So I was talking more about sharing methods for test methods, for example, or even more basic, at a more basic level, scientific reporting standards for what's in the literature. There are standards out there both for preclinical testing and for clinical studies. I mean, reporting standards exist, but I think in the field as a whole, if it followed them, it would benefit everyone, because you can look at the preclinical literature and not be sure what the plan was for what endpoints they were going to look at. I mean, it's just like clinical studies. You can't necessarily know what was planned in advance or how many animals, were sort of omitted from the study at some time. And so I think just at a more basic level, just an understanding so people can read the literature and have a common understanding of that would benefit the field. I'm not suggesting that's necessarily a project for ARM. I think it's a general problem in the field, though, and it's a problem for us. So those are the two things I was really referring to. It's good to share data, your actual data too, but I think that may be a little bit farther. I'm sure that would be valuable, but that's a little farther along because we don't even know. There's no product class. A lot of these things, they're all different. So I'm not sure what we would do with an aggregate data set, which is sort of what I was saying about the MSCs. Well, we get asked all the time, why don't you aggregate your data in this area of product? MSCs is not the only one. I'm just pointing that one out because that's the one where we have this project going on. But people will say, why don't you aggregate your data in this area and see what you've learned? And the thing is, I think it's hard to aggregate it because the products are different, what's collected is different, what people are looking at is different. So it's not really set up. It's certainly not set up for us to aggregate it, and I don't know whether it would be any easier for you all. I mean, because what we see is what you all see. We just see all of it at once.
Mm-hmm. We don't see that.
Yeah, but I can say that it would, even if we had granular data in a certain product area, we'd have trouble assembling it into, and be able to draw a conclusion about a class of things. So it's just hard for me to see how the field could, but I think there's some more limited things about test methods that the industry could look at. If there's a challenge in developing test methods in a certain area, think about developing a standard for that and backing it up with scientific information. And in some sense, I'd make the same kind of comment about the animal testing that, people are always telling us, Ellen, you made the comment about the different requirements from different regulatory agencies. And that's always posed as a regulatory question, but every regulatory question has a scientific question behind it. And the scientific question is, we want to establish safety for this product or this type of product or something. And what test methods have the most predictability in terms of giving us an assurance of safety. And that's not a regulatory question, that's a scientific question. But I think because of the deficiencies in reporting in the literature, and because the scientific community isn't really focused on that, because it's not a basic science question, those questions don't get answered. And so it comes, it gets reflected back to us as a regulatory question, which of course it is. I acknowledge that it's a challenge and it's an added challenge, but I just want to point out that for those type of questions, there is a scientific challenge to the community, and I think the community should consider that.
So, totally agree that the scientific issues have regulatory implications. And so part of it was thinking about there are certain things where one would think the science around assessing tumorigenicity in certain types of cell types might be, there might be a body of evidence that could be acquired, that there could actually be some convergence of guidelines or harmonization, if I might be so bold as to suggest, in particular cell types and tumorigenicity and biodistribution. Those, of course, have to be informed by the scientific basis of why we're making those types of policy or guidance decisions. So part of it, it sounds like on a regulatory basis, you have a venue already with international discussions where you could arrive at a convergence or harmonization, but what you're lacking right now is the data to support going that route. Is that what I'm hearing, or?
Well, I wouldn't say it quite that specifically, but just in general. I mean, we have a preclinical guidance out there, which did the best that we could to assemble our current state of knowledge, and our best recommendations in the field. And if there's more information going forward that would allow us to change it, and we discuss these guidances with the other agencies too. This isn't something we develop, and we share these kinds of things. So if more information came along that made it clear which, what were the best ways to do this, then we would certainly be thinking about what it meant to us and discussing with the regulatory counterparts also.
Well, one thing we do as a funding agency, we have the opportunity to seed the field, to ask particular questions. We have a Tools and Technology Initiative to hopefully overcome some obstacles to translation. We have some other initiatives we can put in place. So it's helpful to know what the gaps are that we could, at least as a funding agency, think about internally about, are there ways that we could help accelerate moving answers to some of these questions?
And then the other area this comes into, which Dr. Witten kind of talked about a little, was you talk about test methods. One of the areas we see is actually in raw materials and manufacturing processes, where again a lot of the processes that we're all using involve needing to use materials which aren't available to GMP or pharmacopoeia grade. And so there's some work going on at the moment at the European level to actually look at where there are common raw materials for manufacturing processes, what kind of risk assessment framework could be used, and looking more to actually how we might be able to start sharing those kind of risk assessments for commonly used materials which aren't available to that grade commercially.
I have a question on that. In Europe, for starting an initial clinical trial application, it's country after country after country, and there isn't any harmonization, such as with the FDA. The FDA covers the entire U.S., but in Europe, it's every country, and the standards and requirements for clinical trial application are very different. Do you see harmonization coming about anywhere in the future, or how are they addressing that particular issue? Because in one country they don't ask anything, in another country they have the highest of standards, require everything before you start.
Yeah. So clinical trial authorization is a national competency in the EU, and it will remain a national competency. I mean, everybody should be working to the same basic framework under the European regulation, but I agree with you, there are differences in the way certain countries may be particularly interested in certain aspects. What's coming in now is a new clinical trial regulation, which will come into force in about 2017. And under that, clinical trial review will still be a national competency, but there'll be a single portal. You make your application down, and there'll be a single review time, and you'll get a single set of questions back from the countries. So it's still possible in that framework that a country may decide that they don't want to approve your application. They can still make their own decision, but at least they are working now in a process where they're speaking to each other and coming up with a list of questions when that new regulation comes into force.
Okay.
So Natalie, do we think that'll actually end up taking more time? Because, we go to individual national competent authorities, and some will approve it in 30 days, some take 60. So if we go towards this new process.
Yeah, so the idea was to try and overall bring the review period time down. But you're right, within the current framework, some countries are much faster than others. So in the regulation, they've stipulated what the allowable time frame is for the review. Unfortunately for us and ATMPs, we got a longer review time than they got for other medicinal products. So I think on the whole, I hope that there'll be an increase. The idea was to get an improvement in time, but until we see things going through it, I think it's hard to know. And I think we aren't going to come to a place where some of the quicker agencies like the UK and Belgium, we aren't going to come to a place, I think, where everybody is as quick as we are at assessing applications.
Any questions from the audience? Okay, so we're almost out of time, and so I think I'll end with asking the panel. So I think we've seen a lot of evolution in regulatory frameworks globally, and I guess when we talk about what we would like to see moving forward in regards to what types of guidances we would like to see from not just from the FDA, but from health authorities globally to help us as an industry develop. Open-ended question.
Certainly on our side, something we've thought about is, and the timing remains to be determined, but product-specific guidances with regards to, if you think of, for example, gene therapy, there's so many different types of vectors and different types of products. Bob explained their product, they administer the AAV directly. We take the cells and mix it with the virus ex vivo and re-administer the cells. These are very different kinds of products. So thinking about having guidances, for example, on the preclinical side or even on the clinical side, and I know the agency has started thinking about that. But as products are going to come, hopefully, to BLA and registration, I think it's going to be helpful to see some of that. And one thing that we're concerned about on our side is making sure that ARM and other groups, including patient advocacy groups, can provide their input as well, so that the guidances are not focused too much on one product because of that experience that the agency may have, but that it encompasses multiple stakeholders' views. So.
Yeah, it's not so much guidance. I guess what we've really appreciated as an agency dealing with, working with investigators from academics and from small biotech, is really the opportunity to interact with FDA and to have an ongoing dialogue. And I guess really encouraging that to continue and increase if possible. Because that's actually a very valuable set of discussions that really does, in the long run, save a lot of time in cell therapy development. So I just wanted to emphasize that that's a piece of what we work with that is very, very valued. I think the FDA does a great job with putting out guidances and getting inputs. Maybe, I guess, faster adoption, but I know the process is very long in terms of the different steps that need to be taken. But those are two things that would be helpful in the future.
So I'd highlight the area of looking at some of the manufacturing and quality requirements for these kind of therapies. Specifically at the moment, we're looking at the agencies actually and trying to, in Europe, get some better guidance on comparability. What comparability requirements are, where you may, for example, have to introduce multiple manufacturing sites, either pre-authorization or post-authorization. And also looking at what we call kind of tertiary manufacturing steps. So these are things you may have to do, a clinician may have to do at the site with a product before it's administered. Sometimes those are classed as reconstitution activities, but where they're classed as manufacturing, that means you have to have a manufacturing license for every hospital site that's doing that step. And we've been looking at changing to more of a kind of pyramid-based model of licensing, such that individual hospitals can become accredited to do that step in a much more simple way than they can at the moment.
Yes, I agree with Natalie, and the wish list would be for the manufacturing to put out a guidance document on manufacturing changes or scale-up that are considered major. Those are considered minor or insignificant. And I think that really helped years ago with the monoclonals and even in the blood products division, the types of changes that are considered in those various categories and the type of documentation that's necessary to make those changes, and the review process and the impact is very important, and that'll be very helpful in talking about making those changes to provide enough product during the clinical trials and then post-approval.
Well, I don't think there was a question for me, but I appreciate everybody's comments.
So now we're up against the time, so we have lunch next, but I just want to thank the panel for their time and their information.